So I'm Jan Kurtéyn. I'm head of production here in Gent's radiopharmacy. Ghent is Ghent University Hospital. So if you want to see the facility, I will be very glad to show you around.
Yeah, sure.
So keep in mind the setting. So we are a 20-year-old facility, which we gradually, with chirurgic precision, we modify things, but it's not a state of the art. I would design it differently. But we have so far found always a solution to our problem. So we've started with internal use. So now, since 1 year, we are also supplier of other hospitals. So we have a logistic setup. So we can supply tracers and therapeutics to other facilities. But yeah, we are not a commercial producer. So we are a public hospital. So our main concern is the care for the patients here and the care for the patients of our partners. But we are not blind for collaboration with private companies. Now, today, it's mandatory. It's mandatory. So we go down now, and our laboratory is just on the floor below.
Below, yeah.
Which gives the nice opportunity, to just produce our doses and we send them up. It's a tube system, and so what we produce, our single dose, we can just shoot up. And goes directly into the injection room. So as I told you before, we have been gradually increasing our activity. Initially, the cyclotron laboratory was there, and that was the only laboratory we had. But steadily we were growing. So we have recovered here is a laboratory that we use for the SPECT and PET dosing. So that's here. Kom maar zo, passeer maar. Is here. So this is relatively new. It was an old clean room that we have modified. That is here.
How many cyclotrons you have now?
Is one.
One.
Just one.
18 MeV cyclotrons.
Yes, IBA 18 MeV. If you want to leave your stuff, maybe you can just put in my office.
That's your bike here, Jan.
It's a bike that we use to go around.
Okay.
And so in the morning or in the weekend and the night, the driver, they bring the technetium or the products that we've ordered. They have the generators or the product, and they have access with the badge, so they can, for example, here are the seeds for brachytherapy that they have supplied, they have delivered, and so we can just they can bring it in, and then the operator, the technician from the other side can take the things, can take the products, and can put them in the...
Are you working also with Quadromet with the Samarium?
Samar we did.
The minus 20
Yeah, we did, but it's been a long time, Samarium. I think once in a while, but it's not a routine thing, no. So just this is what we call the SPECT PET laboratory. We are in the process of qualifying this according to PICS regulations. So it's not yet. You will see, because you will enter. I will just ask you to share a lab code to enter. We'll see if I have enough. But we are in transfer. So we have until 2026 to be compliant with PICS regulation. So we are
1st of January, I think, 26 it should be.
Yeah. So
on paper.
Yeah, we'll see. We'll see what happens. So we are working towards. So if I may ask you to just put on a lab coat. So Yes. So this is the first airlock, and then we move to a second airlock here, where we will do in the future some additional gowning to be able to enter into the laboratory zone, which is here, which will then be classified GMP grade C. That is the classification that we want to obtain in this laboratory.
For you, that's your. For the QC lab, you want to have a
no, it's production lab.
Production lab.
Production
yes.
lab, grade
okay.
C. Grade C.
Because
so
in the QC lab you don't need it.
no, that's not in the production facility, so it's here. So what we do here is the reconstitution technetium that's done in this safety cabinet, grade A. We have a second one there for the others. So when we do the dosing for the iodine products, or also the radium, like Sofigo, the treatment, that is done in the other laboratory, but that's the place where we organized. Here is the generator is there. So that's where we daily produce our spec products. In the laboratory, which we reach through this corridor, on the other side is the PET section. So you have also a type 2 safety cabinet there, where we do the dosing, the individual dosing of the PET product. And as I told you, the injection room is just over, and so we have just a tube system where we put our product in, and it's sent directly. Yeah, it's directly sent to the room where the patient is. The product is administered. So this is what we recovered. It was an existing clean room. We have modified it. We have put shielding around. It's concrete shielding for radio protection. We have changed some entrances, some exits. But yeah, this is not the way I would design it. It's much too big, much too much clean room for the activities. So I would never design it like this. I would design it much more compact to keep investment cost lower. I guess in Belgium, more or less, it's about 10,000 euros per square meter for a clean room. So
at least.
Yeah, and that was maybe the price of 5 years ago. So maybe it has even.
No, your last figures I have was the project in Leuven. You are about 15,000.
15. Yeah. So it's expensive, and also operational cost, the maintenance, but energy cost. This is turning all the time, so you have to keep it as low as possible, especially because there is no air recovery in a nuclear facility. It's all fresh air.
Yes, but
in and used air out, so no recirculation.
And that's a matter of discussion, you know, because I have discussions with the inspection about that pharmaceutical, not the FANC, but the pharmaceutical. Because the famous Annex 3. And Annex 3 says if you can validate, for example, that your risk is below the threshold of exposure of the person working, in beta plus we have it, recovery.
Well, you talked about 15,000 euros per square meter. For how many meters sum? For 100 for?
No. When I'm talking there, that's for the square meter, and it's a room. That must be a room of about. So from ton, I should say about 16 square meter.
16
square. About 4 on the 4 or 3.5 on the 4.5.
Yeah.
The space needed to put the hot cells, of course.
Yeah. And whole investment it's about 1 million. 900,000. 60 meter.
16 square meter.
16
square meter. 16. 16. Yes. And your must be about. This is quite standard. About roughly you're about 45 cubic meter there. 42, 45 cubic meter.
Yeah.
But you have to keep in mind in a Class C, depending on the configuration, and in the Class C, you have practically no equipment. That's open space. Yes, you have a table, you have a computer, the minimum needed to work in it, but you are working about 40 renewals of air. Recycling.
Yeah, we have here on the basis of 20.
Of 20, you are.
We are at 20 here in a C. In a B area, it's even higher, just to get the necessary.
To get the class.
Yeah.
But of course, that's quite important. At the time you are having the commissioning of the equipment, at that time you are measuring how much renewals you calculate the HVAC. You are going for the 40, like I said there. Of course, during the commissioning and the automatization, the robotization of your HVAC, if you can go down to 20, 30, of course you do it.
Yeah. Yes. So this is too big. One thing is smaller rooms. You can put several, here is safety cabinets, but you can put several cabinets in the same place. But you have to take into account that some activities cannot be combined. So here, our risk, so don't share with the authorities. The risk is here that one of the studies here, what we do is blood labeling. Can you do blood labeling, which is tissue, in the same room as you do manufacturing of that is not allowed? So we might have to switch areas and dedicate another lab for this activity. So it's some kind of activities will not be allowed in the same room for risk of cross contamination. So that is.
That's mean if for the future project you will do it from scratch, you maybe will do more small hot laboratories for.
Yeah, it's just to see what are the activities that we want to do and are they compatible. Do we prevent enough cross contamination? It's also the flow of personnel, flow of product, flow of raw materials. Waste. That there is no crossing. So that is also something that you have to keep in mind, that there is no risk of this cross-contamination. Flow of things have to be straightforward without too much crossing. So this is, for example, regulatory, and also in terms of cross-contamination is a weak point here. We combine activities that cannot be combined. There's other things. We use a cell for one kind of product, and we use it for another kind of product. So we do it with a short-lived radioisotope combined with a long-lived. What happens if we contaminate the place? Is also something. It's purely from an operational point of view. Can you combine these two?
I see esophageal. Here you're preparing the doses.
Yes, and so you see that we have had a contamination, which then we covered, but yes, these are things.
So on esophageal, what do you do on esophageal here? Are you
treatment?
A last measure of the activity, or?
Yes, it's a measurement. Yeah, we do the measurement. We have to verify the measurement, and afterwards, after the injection, we have to measure the residual activity.
Exactly. Yeah.
And that's why we do this measurement.
Just to validate if what I'm signing, because I'm signing those certs, maybe you saw that. Next, if you see a deviation, you phone me.
Yeah. No, for the moment, everything okay.
Yeah.
So this is the activity that we perform here. It's what we call we call magistrale bereiding. Yeah. In English, definition could be compounding.
Compounding, yes. I don't understand exactly from where it comes.
Yeah, it's a mixture of pharmaceuticals.
Yeah. Yes.
Is it a preparation? It's a debate, because there is a difference in compliance for compounding and preparation.
Exactly.
And preparation is much more strict in PICS regulation than in
compounding.
or in GMP regulation than
Yes.
compounding.
Yes.
Yeah.
Now, with what generator are you working with a UTK?
Yes.
Yes. That's the UTK.
Yes. The
Originally Mallinckrodt, now it's Curium.
Yeah. Correct.
Registered on Curium Belgium.
Смотрите,
Niet Nederland geregistreerd, dat is België. Curium
Ja.
Belgium. Veel problemen in de laatste tijd.
Ah yeah.
I don't know if you had troubles with the UTKs, but about 2 months ago, I think it's about November, December, there were a lot of troubles with them.
Customer complaints or
Customer complaints about the illusion.
Okay.
Troubles with the illusion.
Not that I've heard of.
No.
So, not that I've heard of.
And how do you proceed for the milking of the cow?
Well, it's a relatively old, as you can see, a relatively old safety cabinet, but still perfectly working. And if there's some maintenance, because it's originally Veenstra, so
Veenstra, yes.
So if there's work to do, I do it myself.
Okay.
It's just it's a little logic automation in it. It's no PLC. It's some kind of logic, and it's pneumatic. So it's just a pneumatic system. So this is also something I've developed because it was broken. So it's going to be a little. It's just a little compressor.
Yes.
That we've built to pressurize because we could not connect to the compressed air of the building. So I've just created a little. I've just made a little connection.
Mobile unit
Yes,
to have
exactly.
the pressure.
And it will come up automatically. So it's the first thing the operator does. Put on the pressure. And then when the pressure is high enough, it will execute my command.
And you can play it to all kind of, because you have the Texas. Of course, Texas is not sold at the time being, but you can use it for the Texas too.
I suppose. It was in the past.
Because the concept of the generator is quite different between the Texas Sys-Bio and the UTK there.
Mm-hmm. So it's coming up slowly. It's because I didn't build up the pressure before. So like this. And then,
because that's heavy.
Yeah, and I didn't build up the pressure before. Normally, as the pressure build up before, then it goes a little bit quicker.
And in the good old times, you see that's a laugh, okay, and you should be at the place where the solution is at a class A. But in the good old times, they put the generator on the table, some LED blocks around, and it was eluted that way in the northern part.
Mm-hmm. Yes. Well, I think it's a practice that is still done in some smaller facilities.
Well, I did it that way when I got my education in 95, 96 as radio pharmacist. The illusion. You say,
Okay.
Dat was niet een lafzin.
Good. Let's go.
Good. We can leave from here.
Oei. Jan.
We have to leave it.
You can just hold it because we will enter another laboratory.
Jan.
So, in between two labs, we have some logistic rooms. I can show you. Nothing very special. So this is the room that we use for the release. So it's in this place. It's just an office room where all the batch dossiers, all the batch records, they come in and the radiopharmacist releases.
It's for quality control.
For it's in fact
for GMP.
It's GMP. Yeah, it's GMP. It's where the radiopharmacist releases the batch. It's just this, nothing special. And so this room we use as a room for incoming materials. So all the materials that we receive, they enter first this room. So we do a verification. If the material is the correct thing that we've ordered, then we put it here. Is the first stage of GMP production, where we will here verify. So we record them, we show that we have put them in stock, and we will do a first quality control check on every item according to a specification. So on all the raw materials we have defined specification, and here QC verifies if they are.
And this storage, it's enough for your needs, or you would like to have bigger?
You see that we have to store things on top of shelves. Never big enough. It's never big enough.
It's
and you increase
Yeah.
is like office space is never big enough. It's storage place. In the beginning it's big enough, but then you see gradually increasing. So yeah, it's if I would have space more, it would be useful, but it's already something that
you have a qualified fridge. I see
the fridges are qualified. So this is the -80, the -20, and then just the 2 to 8 degrees, and the rest is verified. Yeah, it's temperature control. So you have a temperature control on all the.
Also for the inspection, you have to show the qualification plan and the determination of the hottest place in the fridge.
Yeah, the mapping. Yeah. So we are doing mapping here. Yeah, you will see we have replacement fridges, and we are doing mapping before we can use them. Yeah. Het merkt dat de publieke aanbesteding
gekozen heeft.
And then this, the room there is our logistic room for packaging. So we are not a commercial vendor. We are partnering with other hospitals, so it's not very big. You have cyclotron facilities where this is the biggest part, and where you would have at the entrance of all the trucks that would leave. For us, it's relatively small, but we have our systems for.
You're determining the TI transport index here.
Transportation index on 1 meter. That is down here. So I have the availability to supply PET products, and these are the PET. We have 2 kinds, the old and the new ones. So these are the new ones, the new PET LED pots that we have.
Did you buy that in Gent, where you have the local producer there?
Fortal.
Yeah, Fortal, huh? Yeah.
Yes.
Quite compact and not too expensive.
And we have developed together with that supplier, with Fortal, we have developed this one. And this is the type A flight case for the lutetium. Because we produce
small bottles.
Well, it's not bottles. No. In fact, no. We are producing lutetium directly in syringes, ready for use.
Ready for use. Yes.
And so we have this, and so the syringe with the shield goes in, and so we've developed it together with the producer of this, and so we can supply lutetium directly for use.
1 syringe for 1 patient.
Exactly.
Yeah.
Okay. Yeah.
One dose.
One dose, 1 syringe, and we can.
That's where you have here 0.8 curie.
0.8.
No? It's usual for 1 patient 0.2.
Yeah, I'm used to the gigabecquerels. It's 7.4 gigas.
Yeah, 7.4.
Is currently known as the standard dose for lutetium, but we'll see. In the future, we might do some more individual patient dose. So some more. So for the moment, 7.4 is the standard dose for lutetium PSMA. What is the risk? is it worthwhile? There's enough capacity. And capacity is generated not by the companies producing, but by the cameras, requiring less dose.
About cameras, I learned that there is no lobbying to have an extension of the number of PET cameras, to have a revision of the law of 14, to have to increase the number of PET cameras.
Well, Belgium has to.
That's information I got from Dominique van der Ginsten.
Yeah.
You know Dominique?
Yeah, I know. It's to see. There's going to be elections soon, and Belgium will have to save 27 billion in 5 years.
Yes, but it's on the Walloon side.
Oh, okay. Not on PET and not on nuclear medicine. Well, good. Good.
Yeah.
Okay, so a little bit logistic rooms. Here you see a second lock, so where the product is delivered. And lutetium is not yet delivered. Tomorrow we do a production of lutetium PSMA. It's not yet delivered. So tomorrow morning will be
How many
delivered.
patients do you have for lutetium?
Tomorrow we have 2.
Lutetium?
2, yes. Tomorrow 2. It's only internal. The week after we have patient for here and for Bruges, where we supply. It's 2. Well, we have 4 rooms, but we share them together with radiotherapy. So we have 2 rooms, and they have 2 rooms. So yeah, it depends. It's 2 patients per week up to 4.
And during the year, how much patients you have summer average?
We are building this activity, so it's going to increase. I believe last year when we've started, we have seen about 50 patients. So I don't know yet where we are going to be. What else our prospect of this year will be? Something
Because
2 per year, 100, something like this. Yeah.
There is also some. There are some changes coming up. I know that from Novartis with the Pluvicto that is coming. Now it's going to start in April. Pluvicto. Lutetium. That's one of the PSMA lutetiums.
The 670.
And they foresee, for Belgium and the Netherlands, they foresee to have about 4 to 5 patients a day.
Yeah.
That's because also up to now its indication is restricted to the, let's say, to
end of life treatment.
Treatment, and really at stage 4 or 5. I think Dmitry knows that much better than me. That stage of prostate cancer, but they want to have it at the early stage, as soon, let's say, when at stage 1 or 2.
Yeah.
That means that you are going to expand the number of patients.
Yeah. But clinical trials are still going.
And plus the clinical trials that are running.
Still running, so not yet decided. Yeah. So this part is built 3 years ago. So it's the newest part, and is an extension.
Like an annex. It's like an annex. Or?
Yes. It was previously an office building, and then later we transferred it. We transformed it into the laboratory as, let's say, an intermediate, because we wanted a GMP compliant production, but that will take much more time to accomplish. So we've started in this laboratory to quickly start up and satisfy the demands, internal demands here. But we want to develop it as a fully compliant GMP production, but that is future. We don't have yet the budget for it, so it's temporal situation, intermediate situation here.
Хочешь доставать, да? Ну, получше.
It's cold because it's too warm.
the battery is taking
It's process.
a lot of.
You're making your own reportage?
No. I'm part of the team.
Part of the film crew.
Yeah.
Very well. We don't have to really enter the laboratory, I guess, so we can just take the quick technical entrance. So this is just a changing room. We've created at the same time with the laboratory a changing facility, so we could change. So in GMP, no longer allowed to enter with the normal clothes. So all the operators they change here into the undergowning.
You're changing you go to the undergowning.
Yep.
Yes.
Yeah. So we remove our outdoor clothing and we put our pajamas, our under.
But you know the discussion is, but still in Beta Plus, there's a discussion about that because there's a strong opposition to change the underclothes, to work with the side underclothes. Your let's say your bourgeois underclothes, you have to live there and you have to change on those of the side. That is basic requirement now.
Yes, you cannot not enter a B area with and not even a C area with the outdoor clothes. Yeah. So we change here. So we with the laboratory we've created for women and men the entrance to enter the laboratory there, but also the laboratory GMP laboratory over there for the cycle.
We are entering into no gender door.
Yes, technical. It's only for things, but we just we don't cross the line, let's say, and that is again for only people access. So, but I can just quickly. It's basically a similar setup as you have seen in the other lab. So we have an airlock, and we enter here. This is designed to be a D area.
That's a D.
A D area with a safety cabinet where we do the production. Inside you will see a synthesizer. You might not see it now, but it's a synthesizer. It's a Trasys all-in-one, which we use for the lutetium PSMA production. So this is the production laboratory.
Lutetium only here.
Is lutetium and gallium. So the gallium generator is just behind the corner. But we produce lutetium PSMA. We have gallium PSMA. We don't do because we have the fluorine, but we can do the gallium dotatate.
Yes,
that is productions that.
And you still work with a gallium generator.
Gallium generator. Yes.
Not cyclotron produced.
Not cyclotron produced. No. There is a project. There is an idea about producing gallium with cyclotron. We have studied the fixed target, so the liquid. We have studied the liquid, and we are now studying the hybrid target.
The hybrid. Yes, the hybrid.
Yeah, there's a hybrid one, which is irradiation on a solid target. Then an in situ dissolving of the produced gallium and transferring the solution.
It's your own project?
No, it's not our project. It has been developed by a person from I'm not sure. I have to look it up.
I think
it's commercialized by a German company.
I saw publications about that.
It's possible. Yeah, it's possible. Yeah. So this is where we do the production.
You are going from your
So in
the street clothes, you go to a D dress to work there.
Yeah. So what we do here is the gowning in undergowning. Yeah. But for radio protection, the operator working there and there with the radioactivity will wear a complete diving. Yeah. So we are overdressed in GMP, but it's a matter of radio protection, how we are going to dress down there. Here is the QC lab. So it's the equipment for the quality control. So you have TLC, HPLC. We do the endotoxin test. pH is measured on a strip. The Yeah, this is the incubators for the sterility test and the environmental monitoring. And that's about it for the quality control laboratory. So designed as a grade D, and so we would work in aseptic production in an A zone surrounded by a D area. So this will not pass GMP inspection, it will not pass a PICS inspection. So this is a temporal situation which will then transfer to the laboratory, which we have to redefine.
And you have here air measurements.
Yes, pressure measurement to keep the balance.
And particles as well.
Particles, it's not a network. It's not a fixed. We work with independent particle monitors.
These small tubes, it's for pressure control.
Pressure monitoring.
Pressure,
Yes.
With the recommendation is that you should not have more than 1 meter of distance between the sensor and the
detector.
The place where you take it. Where are you meeting that here?
Yes.
Okay.
Okay. Yes. I say yes with a little bit smiley face because it looks like it's 1 meter on one part, and I know it isn't because I've put it. I know it's a little bit more, but you don't see it. It's in a hot cell situation where it's not designed like this, so you have the point of inlet and the point of measurement, which looks like it is 1 meter, but inside the tube goes like this. So, but okay. If I get a question by Van Linden in some week, I say, look, it's 1 meter.
Yeah. But Jan, when I
have it on tape.
So here, the finishing that you have here, this area that's not a D class. That's
the airlock going to the D.
Yes.
Yeah. Now it would pass a D area.
You know why I'm putting those questions? Because I just I still have to write the report, audit of the Almaty facility in the National Institute of Physics, and they are working with doors where you have such the plastic doors, where you have such frame around the glasses, etc. And also you have to work now fully archangé. You have to finish. You may not have all those places where you have cleanability, etc.
Yeah.
That makes the things very expensive.
Sure. So next step is we will go to the cyclotron facility now. If I just may ask you to wear overshoes.
Yeah. Okay. Danke.
Here you go. I have my laboratory shoes.
Wou krijg je die openen?
Here you go. Alsjeblieft.
Let me help you.
Yeah?
You start this one and I will try to do this one.
Ok, j'ai trouvé. Voilà. Hop. Hop là. Merci.
If I may just have one name, the person who will...
Who's going to be radiated?
Well, he's the one
it's not marriage.
He's going to record the company. Exactly, I will fill the rest, it's okay. At the end of the meeting. So we'll just start 140.8. So if you just hold on to this one during the visit, there is no risk. It's just from a nuclear perspective, the area here is under another license. So it's more strict license because it's with the cyclotron. So these are the rules that we have agreed on with the agency. The fridge and freezer here are temporal because we are replacing the other ones. So it's in a contract and we have a replacement once in a while. They're here just in test. So we are doing a mapping on these ones before putting them in place. So I've shown you the area where we stored the materials, and so all the materials there are in quarantine until they are released for use. And once they are released for use, we put them here in our storage, where all the raw materials that we can use are stored. So here in the cabinets, everything should have a label which says released for use, for example. So the QC responsible or his or the QA responsible or his co-worker has
that the kind of things that the inspector is watching, of course.
Yeah. So everything released here, and so here we assemble our what we call our kits. So for FDG, for example, these are kits for the manufacturing, the synthesis of the FDG, the dispensing of the FDG, and the other products, if necessary. They are assembled here.
Ja, werkt nog met een papiersysteem.
Ja. Ja.
Это центеры.
And so also again, the design is a little bit confusing, but it has been designed like this. We have a clean room there and we have a clean room there. Two separate clean rooms. The disadvantage is expensive in building, better one big one. The good thing is now that we can work in this one and have this one in maintenance or even in modification. So this is the clean room that we will in the future modify to satisfy the GMP regulations for the manufacturing of the therapeutics, so the lutetiums and others. We will create there a new production line for this production to transfer the activities from there to here, and we will create there then one big additional QC laboratory. So
that are the old
Yes.
maybe I can show you where the cyclotron is. So from this station we control the cyclotron. We control it also from other parts in the facility. In theory, we could control it. I could control it at home, but this is one of the clients for the cyclotron. The technical area here is where all the power and the steering of the cyclotron. So you see, it's very compact for a modern cyclotron. Here is the technical area. So there is the bunker. We have a plug door, and so the door opens until the end to get us entrance to access to the bunker and to the cyclotron. And here is a station where we do the maintenance of parts of the cyclotron. So that's here. are back where we were. We did a little tour. Oop.
Yeah. That are the rails of the tramways of Gent that you have.
Yes, I stole them. Tram 1. Behind you we have our packaging room. So here is the other side of the production lab.
Yep.
And so here we package our products. So they come out of the dispensing cell, which is on the other side.
Yeah.
And here we package in big lead pots, smaller lead pots, intermediate lead pots, that with an adapter that we've developed, we can fill vials in all kinds of lead pots for the transport of the product.
And the exhaust is linked to the filling cell, where the dispensing cell. Yes, it's about the same concept as what you have in Beta Plus, except that here you're outside. You're getting the closed pots outside. You can say it is a technical area.
Yes, it's a production area, but it's not a GMP controlled area. Well, it is GMP controlled, but it's not a clean room grade. It's non-controlled in terms of grading, GMP grading.
Again, in going back to the concept of the last radiopharmacy built in Brussels, there you have it's coming. The pots, it's about that's another system. It's a common chair system, but you have the filled pots coming on the technical area. But the technical area is finished Class D, but it's not declared Class D.
Ah, okay.
In order to reduce the maintenance cost.
Yeah, of course. Yes. So that is here. All our synthesis hot cells, they feed into this one dispensing cell. Yeah. Which makes it a very, it's like a point which is very, very densely used. So it's indeed bottleneck. Yeah. So we have to, and that's one of the things that we want to create one or even two other dispensers in the other lab to avoid this bottleneck. You cannot imagine daily, because we use this daily for FDG, so we have your daily production of FDG, that you contaminate it with lutetium and you cannot work in it for some time. It's not possible. No, so this cross-contamination is...
You need separate lines there.
You need separate lines. But for the moment everything feeds into this one. Yes, that's our current situation. We have 5 synthesis hot cells and only one dispenser cell. So from here you can have a view inside the clean room. Is the one of the 2 clean rooms. So we are in the technical side. We have 3 Comature MIP synthesizers, and this is the technical side, and inside is the production side, the clean room side. And so we have one. I can open this lead shielding. And for example, here inside is one of the Sintra systems, which is a non-cassette based system. It is an advantage to have non-cassette systems if you are developing things. For pure routine use, cassette-based systems are preferable, in my perspective.
It's Sintra RN Plus?
Sintra RN Plus, exactly.
Do you have service support from?
No. We do it all ourselves. Yeah, why? We do it ourselves. It's a decision. What you get, what value do you get? So we are not a commercial supplier. I would not choose this system to produce routine large quantity amounts. we have time to do it, and we also have the knowledge to do it ourselves, and so we can combine it. So it's just a value for the money. So I prefer to invest in people rather than in contracts for this production line. Yes, we have a contract for the other synthesizers, which we are using for the daily production of the pharmaceuticals. So it's just a selection based on value.
Yeah.
Yeah.
And now the consumables and reagents that you need for preparing the dispensing solutions and the dispensing operations, you bring them in. From where do you have a MAL, material access system, or are you bringing it in?
No, we have the material analog systems. So we have this pass boxes that we use for the materials, and we have our process for disinfection.
Okay.
The disinfection of materials to bring them from a non-controlled from our storage room into the grade A laminar airflow. So it's a gradual approach. It's disinfection, passing through the pass boxes another disinfection to pass until the grade A.
The class C, where you're going to the manipulation, and you go to a B, you have even a class B.
Yes, to bring into the A zone. Yes.
So if you need to change filters, you're doing from this part, you're just changing it here.
The filters for the inlet air of the clean rooms,
from,
you mean? Or
for cell outlet and from inlet for clean rooms?
Yeah, well, the clean rooms, it's endpoint filtration, so it has to be done from the clean room side, to replace the filters. Rather than, we don't do periodical replacement. We do a verification. If they satisfy, so the filter integrity is confirmed, we do not change them.
You have to check once a year.
And how you do
Two times per year.
2 times a year, yeah.
How you do this verification?
Verification, so we have a contract with a company that comes and do the verification according to the ISO.
With what company are you working?
We are working, well, it has changed recently. Before we used CME.
Yes, CME.
Now it has been, since January 1st has been changed to PMT. Yeah, PMT. They also do the requalification of the LOFs here. Yeah.
So they do the global GMP equivalent required. Yes.
Yep. According to GMP. So we don't do it ourselves. We have a company that does this verification. For the A and B, it's 2 times per year, and for the C and D area, it's once per year.
Yes.
Yeah. We also do internal tests, so all the microbiological testing we do ourselves.
Also yourself.
Yeah, and in some areas we do also. So they do the tests at rest. We do the tests in activity.
Yeah. And for the B, the B area. Don't you have once a week after cleaning, monitoring, microbiological monitoring?
Well, yeah, there is periodical monitoring that we do.
The basic one very complete, and
we have the
reduced one.
Yeah, which with every production we have our environmental monitoring.
Yes, of course, you have the monitoring at production.
During production.
That's mandatory.
Yeah. So surface control, air control, particle control, clothing control. So we also have the fingerprints and the it's a rotation that we do to verify the gowning. Yes, that's for our B area. C is less strict, of course. There is just periodical controls. Yeah. Yes.
Let's go.
Go. Yep. I will maybe just close it, then I don't get trouble tomorrow with my colleagues that cannot transfer the activity because somebody left the door open from the technical part. So we have two more Sintras there, and on the first one we have the carbon 11 system, which is a GE Tracer Lab FX. And inside the other clean room we have two hot cells. One is equipped with a Sintera and the other is equipped with a Sintera with a loader, an extension and a preparative HPLC.
And the class C, they are working in under pressure or in overpressure?
Overpressure.
Overpressure.
Yes, yes. You go from, so this is at 0, let's say, the common area is 0, and then you will gradually go up by 10 Pascal. Nothing to worry about, it's just an indication that the battery is out. Apologies.
You know the concept that UZ Leuven we are working there, the nuclear area is in under pressure, is -10. Complete area where there's a nuclear risk, but the class C there, that's a big one, but it's split it in two smaller ones. There we devised it at +30. So it means, of course, that you have to go through before you go in the C through the D. You have steps, otherwise you cannot open the doors. But the approach also in Jetten, Brussels, the contaminated area is in under pressure. That's the way I know working on the project that we're at Almaty.
Normally, it's the same that we did in the other labs, is a SAS, an airlock which is in under pressure, that all the air goes in.
You can work like
outside.
that also, that's possible.
Or a surrounding area. So here we have one of the two QC labs in our cyclotron facility. pH probe, conductivity meter, that's the gas chromatograph for residual solvents, and one of the HPLCs with the auto sampler. So we are running tests for the moment on this one. And then we go out through the other side, up. And here as well, we have our first QC lab, also equipped with HPLC system, TLC system, endotoxin tester, workstation, analytical balance, and the necessary storage places.
This one is TLC?
Is a TLC scanner, yes.
It's a new one?
It's a new one, it's last year that we bought.
Okay.
Yes. All right. And so this is the first clean room here, and the second. So we have been at the technical side of that clean room. Of this clean room, we have been in the packaging where we get the drawer. And so this is, for example, a modification that we have done. We have, with the approval of the agency, done a modification. We have created an extra barrier. So we could go from a non-controlled area through this intermediate area into the airlock that gives access to the C zone, and behind the C zone, so then after this airlock you enter into a C, class C. From the C area you can enter in a final airlock that gives us access to the B area for the
You also added ventilation, or you made just a curtain?
That was, it's a curtain, and the ventilation we changed the position, so it's just a little bit pulse. It was somewhere here, and I've just made it there, so we have some kind of flow. So it's a little bit what we call bric-a-brac, but was accepted by the by the agency as a solution for.
That's a laugh there. A PAL. That's a personal access there. Yes.
Yeah.
And then you come with the dress that we have here. Yeah. The side dress.
Well, we will not enter. We come with our undergowning.
Ah, okay.
In the first airlock, we will put coverall.
Yes.
I can show you. It's here. A non-sterile coverall. There we put.
Yeah. Yes.
To enter the sea area.
Sea area. Yeah.
In the other airlock, personal entrance. There we will add a sterile coverall with a hood with boots, with a sterile face mask, with the sterile gloves, and also, but we have not yet started the sterile goggle. Yes, that's what it's in the final area. So.
And you're working with disposable.
It's a temporal situation because, as I told, we are a public. We are a public institution, and we have changed our contract. And it's now we are waiting 1.5 years because every time there is something new. And now recently, apparently, we are in the phase of the decision of who is going to supply. So we are in a temporal situation where we use disposables because this takes a lot of time.
No, I'm talking about the Class C dress, reusable, but you have to be careful when you are electing the company. With Elis, you remember we had a lot of troubles with Elis.
Now there are two in the running, 2 companies, but there is a globalization. Before you have several small ones, and now you have 2 big ones.
2 big ones. Elis and I didn't follow that.
The Elis group, Elis group, and the other one is the name escapes. I can look it up.
It's not important at this time.
Thank you, Jan, for this great opportunity.
Oh, you're welcome.
To look at this nice site, and I think our colleagues from Teranostics and from Teranostics Association also appreciate you for this opportunity. Thank you very much.
You're welcome. Thank you.